Hello, welcome all, and thank you so much for joining us today to learn about applications of BioMakers' unique BeCrop technology. BioMakers is a global agtech company utilizing next-generation sequencing and a proprietary bioinformatics pipeline in order to provide actionable insights in biodiversity, nutrient cycling pathways, as well as crop diseases, which will be a topic of today. We will cover each of these three different insights that BioMakers provides in a webinar series, and today we will be focusing on the disease risk, as I mentioned. I would also like to take this opportunity to welcome our BeCrop advisors. If anyone else is interested in becoming a BeCrop advisor and utilizing BeCrop technology for your clients, look out for a follow-up email with the information for that, as well as instructions to receive your CEU credits if that applies to you. Today we have Jacob Parnell as our panelist here. Welcome, Jacob, and thank you for joining. He will present a very interesting case on Fusarium and bananas, followed by a Q&A at the end. So save your questions, put them in the chat, and we'll get to them. Jacob, please give us a quick intro, and then we can go right ahead with the very interesting case study.
Sure. My name is Jacob Parnell. I received my PhD from Michigan State University, and I'm a microbial ecologist, but I've really been looking at microbiomes in agriculture for the last 8 years now. Part of it's been in research organizations. I've been in industry as well, and right now I've been about a year in bio. In fact, I think today is my 1-year anniversary of being at BioMakers. And so one of the things that I do is I try to take the sequencing information, and we really try to make it something that everybody can use. There's a big chasm, a big, between all of the data and actually using it usefully and trying to put some application behind it. And so I'm going to go through an example of this is a real world example that we used. So I'm just going to share that, and hopefully everybody can see my screen. And so really what we try to do is like this slide says, we try to decode soil biology. It's a little bit complicated, but it doesn't have to be. And so that's really what BioMakers tries to do, is they try to take something that can be fairly complex and really distill it down to something where we can actually bring it out to the field to use and to make agriculture better, make soil health better. And so this particular study was one that we did in the Philippines, and it was a large study that we did looking at Fusarium in bananas. So this is what the kind of trial design looked like. There were several different areas that had kind of banana problems, and the severity of infestation of Fusarium that we're looking at, this is what the growers noticed. So there were some areas where they said we don't see any Fusarium infection in these plants in these areas. We see a little bit in some other areas. Some have moderate and some are severe. So this is just based on the phenotype of the plant itself. And this is basically kind of the size of the area that they were looking at. And so they collected samples according to those sizes, and they sent those samples to us, where we were able to do the DNA sequencing of all of these samples, and then go through and look for basically the biology of those soils to really try and figure out from a biology standpoint what is happening in these soils. So this is one of the first things that we noticed. This is looking at alpha diversity. So really this is just looking at each sample individually, and it's saying, how many different species are there So that's one of the metrics of alpha diversity. And another metric of alpha diversity that we look at is how distributed are those species. So we could have 100 species, but 99% of it is one species, and then it's really kind of skewed toward that one species. And so as far as the evenness goes, that's going to be quite a bit different than something that is more kind of uniformly distributed across a wide variety of species. So the one thing that we saw here, this is a Shannon diversity index. So this looks at both the richness as well as the evenness. And one thing that we realized is and in this case, I'm going to go back one slide just to point out that we had quite a disparity in the number of samples that we collected. And so some of the examples that we're looking at are going to be between slight and 0, just so that we can basically we're comparing the same number of samples. And that's what we're doing here. So we're really just comparing the slight and the 0. And one thing that we saw is that there's a significant difference in the diversity of bacteria and archaea. So the areas with no visible Fusarium stress had significantly lower bacterial diversity than areas with slight fungal or Fusarium infestation. And then, so this is looking at bacteria. If we look now at the fungal, we're going to kind of focus on this graph over here, where we're seeing we're getting, it depends on what you're calling your p-value cutoff, but we're very close to significant, where we're seeing that we have increased fungal richness in these areas that have 0 Fusarium. So where we don't see Fusarium, we see lower bacteria and higher fungus. Now, those of you who understand kind of the fungal to bacterial ratio, that's really important, kind of a descriptor or an index, a measurement of a soil health. And so one of the things that we're seeing is that with lower bacteria and higher fungus, these areas with 0 disease pressure, let's say, have a much better fungal to bacterial ratio than this other one. So there is something going on here in these soils where we can see that this measurement, this index, is showing that we have a benefit in just the microbiome of these areas with 0 Fusarium indication, 0 Fusarium presence in the crop itself. So that's kind of one of the first things that we looked at. Now, one of the other things that we wanted to look at is the actual presence of Fusarium itself. Since we're going through and we're sequencing all of the microbes, the fungus, bacteria, archaea, all of those organisms from these soils, we're able to actually look at the actual Fusarium presence. And we can see that so I've got it divided out into kind of moderate here, severe, slight, and no fungal or no Fusarium. And looking across these samples, we don't really see a difference between them. We don't see that the areas with severe and moderate Fusarium have a whole lot more. I mean, there is quite a bit of Fusarium, but it's everywhere. So it's even in the areas where it's 0, it actually looks a little bit higher than areas where it might be in severe. And this is kind of just another visualization of that across kind of some of these to look at how this changes. There is no significant difference between these areas with moderate and severe and these areas with none that we're able to look at right now. So essentially what this is telling us is it's saying that it's not that we're seeing more Fusarium in those areas and that's what's causing the problem. There's potentially some underlying problem in those areas. So as we start to look at a couple of other things, so one of the things that we found is that comparing areas with no fungal manifestation, no Fusarium manifestation in the plants versus moderate to severe, we saw that there is a significant increase in antifungal pathways. So these are going to be pathways that are natural in bacteria and fungus that are antifungal to other fungal organisms. And so we do see that we've got a significant difference. And so the way that we've presented this, this is called a BeCrop score. And what this is in this case, we're looking at a banana crop. And so as we place the samples that we've collected in this study, kind of the number of antifungal pathways that we have across all of the antifungal pathways in bananas that we've had. This is basically this sample would show up as kind of a low sample in the lower, in this case, it's at that 36th percentile mark. So it's fairly low. And then these ones are showing up as the high. These are the highest we've ever seen. These are the 100% kind of along that scale of all of the banana samples we have ever looked at. And so really, and that's kind of how we've measured these scores. And so really you can see that there's quite a bit of a difference in just some of these antifungal pathways. So even though we've got Fusarium that's about the same level in these two, we see that there's the community to, I guess, to fight Fusarium is much stronger in these samples, these areas that have no manifestation in the plants themselves. Another thing that we saw that was fairly interesting is, so these are our samples again of 0, this where we didn't see any Fusarium. Here's where we saw a little bit of Fusarium pressure, and then severe and moderate. And one thing that we notice is this is kind of along a timeline as we're going through kind of the sampling sequence. And we saw that in both cases, in this case around towards the beginning of January when they collected samples, there was a huge spike in Pseudomonas. That spike was anywhere from 25 to 30% of the total bacterial population was comprised of Pseudomonas. Now, going back through some of the literature, we see that some of these Pseudomonas strains do have some biocontrol properties against organisms like Fusarium wilt. And so one of the things that we might be seeing is that in these areas where we don't see any of the manifestation of the Fusarium in these crops, we have something that's showing up fairly early to really fight off the Fusarium itself in these areas where we don't see it. We don't see it in this moderate and severe areas. And so these are just some interesting insights into kind of what the biology is doing over time in each one of these different spaces. And so one of the things that might be interesting is kind of an approach to really solve this is looking at maybe what happens if we put we inoculate or spray crops with a Pseudomonas, a biological, around the same time that we see this spike naturally occurring, because we don't see it naturally occurring in these other areas. And so if we can artificially create that spike, that might be a timing where it might disrupt the Fusarium, and we can maybe prevent Fusarium from happening in these other two areas. And then one last observation. This is a general stress adaptation pathway. So this is looking at a suite of microbial pathways that are designed to basically help the plant endure stress, either abiotic or biotic stress. And we see that in these areas where we've got none of the Fusarium that's showing up, we actually see that we've got basically an 11 BeCrop score increase, and it is significant over these areas where we've got moderate to severe. So what some of this is telling me is that kind of as a as an overview of everything that we're seeing is that it's not so much that you have more Fusarium in these other areas. Based on some kind of this bacterial to fungal ratio that we see, that difference based on differences in, you know, the stress pathways in the natural fungal control that is there in the microbial population, the areas that are experiencing more of the Fusarium manifestation in the crop, it's more because those areas are more stressed. The soil health is poorer, the plants are more stressed, and so then they become more susceptible to the Fusarium that is there. So kind of if we take a step back and think about treatment for a little bit, it might, you know, people might look at it and say, well, we need to spray fungicides. We need to spray something to really keep down the fungal population in those areas that are severe and moderately infected. And that could probably help a little bit, but really the problem is that those soils, the plants are still going to be stressed. And so probably the better approach is to go through the biology of the soil and say we need to find a way to work it better. And so a lot of these other things that we've seen, you know, the higher antifungal pathways, the spike in Pseudomonas, all of these areas, these are kind of biological indicators that one area is naturally able to fight off the Fusarium better than the other area. And so it might be better to kind of focus on how we can do that. So I've got a couple of potential recommendations. So like I said, based on that bacteria to fungal ratio, lower fungicide, lower stress, it might be a good idea to use some regenerative approaches to those soils to build the soil health in those areas. If you're going to do applications that are you know, biological fungicides like the Pseudomonas that we were looking at, or look at biostimulants as potentially something where you're starting to build up the soil health rather than just kill off the Fusarium. Yeah, and then like I mentioned, you know, maybe look at a Pseudomonas that you could apply at those key times where we see it naturally occurring in those other areas. So this is just a real world example of one way that we can go through. We can look at the biology and we can really see what's happening in these soils and try to use the biology to inform and to give recommendations to growers. So Ava, I think I'm going to turn the time back over to you now. Is that right?
Sure. Thank you so much for all that very interesting case, seeing that it's not so much the level of Fusarium that's creating more disease in one area, but it's everything else, and how at Biomakers we really can look at and provide insights on the everything else that is sort of really important to take into account when making decisions. So thank you so much for that. And so with that, we're going to jump into some questions over here. One question we have is, can this strategy be used for other crops?
Yeah, that's a great question, and absolutely. We have tested, I think we're up to about 150 different crops now. And so we've got that, you know, the database built of thousands, tens of thousands of samples. And so we can basically do the same approach to basically any crop. Well, any of the 150 crops. And so I think it would be, I would actually be interested to see what major crops there are that we haven't done this with, or that we don't have this background with. So yes, we can definitely do it with other crops.
Wonderful, thank you for that. How does BeCrop measure soil biodiversity and functionality?
That's another excellent question. So Biodiversity is really looking at the different species. And so it's either different species spatially or temporally, or within two different samples. And so the way that we look at it is we can look at the biodiversity of one sample by just going through and sequencing and figuring out how many different species are in that one sample. Fungal species, bacterial species, and then, like I said before, so evenness is another one, and that's just that distribution of species. And so we can get an abundance of these species. So we have our approach allows us to do, it's semi-quantitative in which we can get a relative abundance of different species. So like I said at the beginning, if we have one species that is dominating the community, 99% of the community is this one species, that's not going to be a very healthy community, and that's going to show up in that evenness measure of alpha diversity. We can also then compare two different groups and say, how much overlap is there between these two? Or if for example, if we are using fungicides, and if I go out and I spray fungicides on this area, how is that impacting the microbial community? Am I killing off a lot of good organisms? And so we're able to look at those areas of biodiversity as well. We look at long-term biodiversity, and we've actually gotten fairly recently. There's a new one that I think we'll probably, I don't know if we're going to be doing a webinar on this yet, but there is a new, well, fairly recent, within the past maybe 2 decades, idea in biodiversity that's called it kind of an interaction diversity. So it's not necessarily just who is there, but how closely are they all interacting, and how closely are they forming as a community? Are they just all individually fighting for their own resources, or have they built like a community that really starts to grow? And that is one of the things that's used in our BeCrop rate to look at kind of these more biodynamic or regenerative systems, is that interaction diversity. And so that's a really key indication of how healthy the soil is how much those interactions are happening. So that's the biodiversity side. The functionality side, once we've got that list of all of the species that are in the soil, we take that back to another huge database that we have, where we've got fully sequenced genomes of thousands and thousands and thousands of microbes. And so we look and we say, okay, we see that we've got, for example, we've got a Pseudomonas in there. We'll call it Pseudomonas aeruginosa. And so we're able to take that back and we say, we know it's in there. We go back to this other database that has the full genome sequence of it, and we say, these are all of the functions that Pseudomonas has. It has antifungal properties. It has some, maybe it has some other PGR, some, maybe some phytohormone type of pathways that are able to interact with the plant quite well. And so we're able to look at all of those different functions that are in there, and then we're able to do it for all of the organisms that are in the list that we have of that soil sample. So from that soil sample, we're able to say, soil sample A has, a whole bunch of Pseudomonas, and so it has more of these antifungal pathways. It has more of these, stress response or phytohormone pathways, or whatever it is, whatever functional pathways. And sample B has, fewer Pseudomonas, and so it has less of that functionality than sample A does. Now, the difference between the biodiversity and the functionality is the biodiversity is constrained by the species. The functionality is constrained by that function. So there are a lot of times when we look at it and we see that the biodiversity of two soils is different. So they have completely different communities. But when we look at the function, they're actually very similar, which just means that, instead of a Pseudomonas that has all of those things, maybe we've got a bacillus in this other community that has some of those similar, or, a makeup of different organisms that have similar function that kind of do the same, play that same role as the Pseudomonas in this other. And so that's basically how we look at at the functionality is looking at all of the genes of all of the organisms that we see in that. And I hope that made sense.
Yes, thank you so much. Another question we have from the audience is, which Pseudomonas species were found in your research?
So for this one, I only went down to the genus level. We can go down to the species level. I don't think I did that here, but you can, once you get this back, you can actually drill down to a species level to try and find some of that information. So you can, in this case, I only looked at the genus level, and I saw, Pseudomonas, that looks interesting. We see this spike, and so, but if you wanted to, you could say, well, instead of looking at the genus level, I want to look at the species level, and you could look at that. And I don't recall if I did that on this one.
Okay, thank you. Next question is, the soils where you took the samples, were they similar soils?
They were, so yes and no. So they were similar in the same way that, corn belt samples are going to be similar, right? So they're in the way that they're like mollisols. In this case, they were, I don't remember what kind of soils they were, but they were kind of the same soil classification group. But there are some biological, some chemical differences in the soil. So the answer is kind of yes and no, in that they're the same generally, but when you get down into more specifics, they are a little bit different. That wasn't a very good answer. I apologize.
No, all good. Thank you. Next question is, how many samples did it take to build your database?
So right now, our database, we've collected well over 35,000 samples, soil samples, and we've sequenced them. We've got all of this information. And so the great thing about this as well is that the more samples that we collect and we add to this, the more refined, the better the questions that we can answer. And so we've gotten, our portals that you would interact with if you're a BCA, the BeCrop portal that you would interact with is constantly being refined to get better and better and better answer the questions that we're looking at as that database grows, as we get more information on what the biology of all of the soils of the world look like. And that's one of the great things that we have is just the amount of, the soils that we have. There are a couple of other companies that do something similar to what we do, but they're so focused on their geography and their crops that that unfortunately they're not able to look at kind of this more global approach to this. And so this is one of the things that Biomakers kind of really stands out.
Awesome, thank you for that answer. Next question is, you mentioned that low Fusarium impacted areas may indicate poor soil health, is stressing the plants. You'd think low Fusarium impact would be due to healthy soils. Can you elaborate on that?
And I may have, so the areas that have higher soil health are going to have lower Fusarium impact. And so I don't know if I misspoke at one point and said it the other way around. Is that the question about that I said that backwards?
Yeah, the question is quoting you.
Okay, I am so sorry. So yeah, if I said that, it's the other way around, where those areas that have those healthy soils, where the plants are not feeling that stress, are the areas where we don't see the Fusarium actually infecting the plants. Now, the interesting thing, remember, is that the Fusarium is pretty much the same all across all of the areas. And so it's not a difference in Fusarium that's causing the problems, it's something else. So I apologize.
Okay, thank you for clarifying that. Next one is, has similar research been conducted in tropical soils, particularly African soils, and couldn't there be varying results?
Oh, absolutely. I think that if you went to another place, maybe you would see that there is another area where you are actually seeing more Fusarium in those areas, or you might see something else. We have done a few. I think we've done a few studies in African soils, and we're talking with people that are doing research in Africa now about doing more studies in African soils in some of those areas. But yeah, I think each one of these studies is going to show something different. Every time that I've looked at one of these, the outcome is a little bit different. This one I thought was interesting because it's not what we thought it was going to be. We thought what we were going to see was that we would see more Fusarium in those areas, and it turns out that it's something else. And so in this case, as if I were a potential BeCrop advisor working with this group, I would say that, yeah, going out and continuing to spray fungicides is not going to really solve the problem. It might keep the population down, but now we've got some clear ways of actually solving the problem rather than just kind of keeping the problem at bay a little bit. And so I think each study that you would do, and I think Ava's going to be able to show us how we would kind of set up each one of these studies here in a little bit. But each one of these trials or these studies that we would do is, potentially has a completely different outcome, and it's all based on the biology.
Awesome.
I think is Ava still on, or
Oh, can you hear me? Sorry.
I lost you for a little bit. I don't know if everybody else.
Okay. Okay, next question is, how did you qualify the infestation severity for the samples used for the study?
Yeah, that is a great question, and that was all. So that was done by actually the people that were on the ground that were collecting the samples. So those were basically the trial owners that said we have a problem, and so they're the ones who basically wrote down on their samples that they submitted to us. They said these are areas where we're seeing just severe Fusarium. These are areas where we see it moderate, and so forth. And so we didn't put those on. So those were something that they put on there. And to be honest, they're going to know a lot more about Fusarium infestation in bananas than I would. And so yeah, those are something where we collaborate with the growers or the landowners, whoever it is, to for them to say these are the problems that I am seeing in the field, and I want to know the difference between these problem areas.
Very good, thank you. Another question is, did you measure differences in mycorrhiza?
So within the BeCrop portal, we are able to look at differences in mycorrhiza. I think I looked at it, and I don't remember if I saw anything in those differences when we looked through that. But that is something that we can easily do in the BeCrop portal. There's there's an explorer tool. It basically looks like a petri dish with a lot of little bubbles. And if you open that for one sample, you can actually click on and say, I want to highlight all of the arbuscular mycorrhiza, and then it'll basically highlight all of the circles, and in each one of those circles, it'll give you an abundance of that particular arbuscular mycorrhizae in the soil. And so through that, you can kind of go through and you can figure out, okay, sample A has this much, sample B has either more or less, and so you can kind of go through and figure out in each one of your samples how much mycorrhizae you have.
Awesome. Thank you for that. Next one, would you ever recommend inoculating soils to account for out of balance bacterial or fungal ratios? For example, using mycorrhizal fungi to balance high bacteria.
So my experience as an ecologist is that that would potentially be a quick temporary solution. So for example, in these soils, you could add adding that spike in Pseudomonas could also be a fairly temporary, but really what you want to do is you want to give the soil an opportunity to balance itself out. So practices that are longer term, more regenerative. So definitely, you know, like I've said here, adding a biostimulant that's going to actually, you know, have more long-term benefits of, you know, maybe your biostimulant is building carbon more, or, you know, liberating more nitrogen or something where you're actually trying to give the soil itself a chance to kind of equilibrate. And that's, you know, again, going back to what I'd stated before about our BeCrop rate, one of the things that the BeCrop rate is really good at doing is it's looking at some of those things that are saying, are my practices actually building that soil health? So the BeCrop rate essentially is a soil health indicator, but it's so advanced and so specific that we can actually, if you just sent us a blind sample, we could say, these are the practices that you are currently doing on that. So you're doing conventional practices, or you're doing somewhere around no-till or organic, or, you know, we can basically tell you based on the soil health what practices are being involved there. And so really what you want to do is you want to push that soil health in the right direction. Sometimes that's going to use biostimulants, but sometimes it's going to be different practices as well.
Awesome, thank you. Another one is, if I understand, you can describe the apparent or putative functionality in the sample, but you cannot yet confirm the functional gene that is engaged. Do you perform any type of metabolomic or metabolic analysis?
So yeah, we don't do any metabolite or metabolomic analyses. And one of the, well, there are a couple of reasons for this. One reason is that metabolites are going to be very fleeting in the soil. Well, some metabolites, some are going to stick around a little bit longer, some are going to be very short, but then trying to build a story out of that is going to be a little bit more difficult. So that's something that would be very good to, for example, with this Pseudomonas, if you were to take this, maybe, you know, take the Pseudomonas that you actually found in these banana fields, isolate it, bring it back into the lab and say, what metabolites is it producing? And is it really, you know, producing some of these antifungal things that we think it is? And, you know, are they different than, so you can kind of go down that research angle there. But we don't do that with our samples because we're looking at longer term. And so most of the metabolites would have disappeared, even, you know, and so a lot of it is just inferred. So if there is a niche in the community for, let's say for denitrifiers, you know, there's a lot of you know, nitrification going on or something going on in the soil, but there are no denitrifiers, then, you know, if that niche opens up, then there is something that's going to be there to fill that niche. And so that's what we're looking for, is we're looking for how have the niches changed? So what are the niches that are in these disease areas versus the not disease areas? How have they changed? And in the case of the disease areas, we're seeing a lot of it has to do with abiotic stress and things like that that are not happening.
Wonderful, thank you for that answer. Next one is a very interesting one. What is the mode of action of Pseudomonas against Fusarium?
I don't know this one, and I don't remember from the paper that I have placed here. But I do, is this presentation going to be made available to all the participants?
So we have the webinar that can be replayed, and we can offline discuss whether or not we can share the presentation itself. But the recording for sure will be available for everyone.
Well, here I've gone back to the slide. So this is the paper where we saw that. And so I do believe that within this paper, they go through and they discuss kind of, it's a combination of the Pseudomonas and Trichoderma that really work with it. But here they go into discussing more the mode of action of these two organisms. So,
all right, wonderful. Thank
you.
you.
The title, and you can look it up.
Awesome. Thank you so much. And while you look that up, maybe we can, and we have their information, so we can contact you, Dr. Rajesh Kumar, and follow up on this question. Moving on, can you use the same testing to identify ratios in compost? And do you have a database on anaerobic versus aerobic species?
Yes, yes, we do look at some of these in compost. We have done quite a bit of like vermicompost and things like that we have looked at, and we look at some of these, the differences in the function of composts. So yes, anything that is soil-like and has, so our platform doesn't work as well in like hydroponics and things like that. It's best in soil. So any soil, sandy soils, compost, high organic matter, pretty much, I think almost any soil, this does a really good job. We're able to look at it. And then, I don't remember, what was the second question, Ava? Or the second part of that question?
The second part of that question was, do you have a database on anaerobic versus aerobic?
Yes, we do. And as you go into the BeCrop portal, so this is where you can go in and you can actually do all of these analyses yourselves on these data. We have aerobic respiration, and we also have fermentation. And so those are kind of our comparing anaerobic versus aerobic in those soils.
Wonderful. Another question, do you have any other sites to reference where farmers are using different methods like cover cropping or specific cover crop blends that are contributing to differences in Fusarium infection?
I don't know of any that are specifically looking at them in the aspect of Fusarium infection. We are working with a lot that are looking at till versus no-till in different cover crops, and basically looking at the biology and from short-term to long-term perspectives, different crops that way, or different types of cover crops, but I don't know of any that are looking at it as specific to Fusarium. I mean, it's not to say that we can't. I just don't think we have.
All right, awesome, awesome. Thank you so much. And oh, we have another one that just came in. Why organic amendment only suppressing to pathogenic microbes? Why they are promoting growth of beneficial microbes? Please consider my question again.
So what was?
I'm not sure if I'm following there. Dr. Rajesh Kumar, let us take this conversation offline so we can maybe have some face-to-face time to address all of your wonderful points and questions directly. I would like to take an opportunity, Jacob, if you would be able to show some of the slides on the proposal portal to talk about sort of how these projects can be put together very quickly. And then maybe we can buy back some time as well after we do that. So the proposal, the project design portal is a portal that we have put together so that BeCrop advisors and anyone else who's interested in working with BeCrop and offering BeCrop technology to their clients can basically create an account and to sort of put the projects together, create different different questions that they want to answer, and the platform itself will generate a proposal for how to for the number of samples, for BeCrop rate, for example. If you wanted to do a BeCrop rate, the platform can calculate the number of samples that you want. Otherwise, if you are looking at BeCrop for applications on nutrient management or disease or just management in general, you can sort of start with the number of acres, and you can look at different crops that we are working with on there as well, and to create a very customized way to answer some very specific questions. We can send you a follow-up email with a link to schedule a demo and to go over this, and we can have someone on one time as well to answer any other questions you may have about the project design portal or the webinar as well, and we can follow up on that. So Jacob, do you have those couple of slides on that we could sort of just give a really quick visual presentation of what that portal looks like, and then we can talk more about it, about the different capabilities and functionalities if you're interested in a demo.
Are those the slides that are on there now?
I think that you did add them, yes.
Okay.
My screen sharing is not allowing me.
Okay, just let me know when I need to advance the slides.
Thank you so much. So this is if you can see what the text says under there. So these are different sizes of projects that we can do with BeCrop for BeCrop trials specifically. So I don't know if you can read it, but you can answer some basic questions, or you can answer very complicated questions. For example, a project could answer the question of, what does my product do in one location? And then you can take that a bit further and then test more locations and see, what does my product do? Or where should I recommend my product, which is the most advanced way of organizing a project to get more samples and more statistical significance that can help us really drive the point home in some of these bigger questions. And if you want to show the next slide, please. So these are some of the ways that you can add different areas, acreages, crops, and then management styles in order to build a project. This information will be inputted by you, the user of the platform, and once you go through all the steps, there will be a proposal generated that you can share with clients or that you can share with your team, and then you can have it will give you an estimate price on the project as well. Next slide, please. So as you can see here, today we're focusing mostly on disease, even though disease and management and yield are all interconnected. We will have 2 more webinars on yield and management, more specifically, focus more on each of those. But the project design portal also allows you to customize your projects based on the questions that you're trying to answer and the angle that you're trying to look at the results from. Next one, please. Next one.
I think that was all.
That might be all of them. That was all of them. So as you can see, this portal makes it really easy to put projects together, to sort of think about the different ways that you can add different insights to the same project, so you can reutilize the same samples to sort of reanalyze them to answer a different question. It'll allow you to do that as well. That's a lot to cover for today, so look out for that invite to schedule a time with me to go over this one-on-one. We could do a demo and to look at how to use all the features for BeCrop test, BeCrop rate, as well as BeCrop trials. And with that, I'm going to take another look at our questions board and see if we have any new ones that have come in.
Can I just mention one thing about this really quick, Ava, as you're looking that up?
Sure.
So as you go through and fill this out, you know, you fill out, you know, your soil, the number of acres and what you're looking for. In the end, it's going to basically tell you how many samples you need to collect. So it's really a cool trial design, so that you don't have to figure out, okay, you know, if somebody has a, you know, 300-acre cornfield, and you know, there's a, you know, 30-acre part of it that just is underperforming, and so that, you know, you put a low yield on that 20 acres. How many samples do I need to? You don't have to figure all that out. This will figure that out for you. And in the end, it'll say, this is how many samples you should collect in the high yielding areas, in the low yielding areas, or in the high disease areas and low disease areas.
Exactly. A very, very a streamlined process for very complicated project design. So do look out for that email link. And I don't think we have any new questions that have come in. So we can buy back about 7 minutes of our time. Thank you so much for joining today. Thank you so much, Jacob, for the very, very interesting presentation. And we will be in touch. Thank you so much, and have a wonderful rest of your day, everyone.